Thank you very much, our panelists. And we will welcome our two. Did somebody fall? No? We welcome our two additional panelists, Dr. Somesh Chattopadhyay, lead mathematical statistician with the Office of Translational Sciences. And we also welcome Dr. William Chong, who's the director of the Office of Safety and Clinical Evaluation within OGD. Okay, everyone. I'm so happy to see everybody still here at the end of the day. Welcome. Online folks, I hope you're hanging out with us as well. Any in-house questions? Acknowledged. Let's go to our online questions, please.
First question for Dr. Betsy Ballard. The taxonomy seems like a complicated way to address a complicated analysis. Does the agency think this is an improvement from a review standpoint compared to what you are seeing in submissions?
I think the purpose of trying to develop something like this is very often the determination by a review team or even an individual reviewer can be very subjective. So it might vary from team to team or from reviewer to reviewer. And if we have a more qualitative type of method to apply, the hope is that the industry will see less of that variation and have a more consistent response in our recommendations.
Thank you. Next question, please.
Great. Next question addressed to either Dr. Ballard or Dr. Streets. A risk-based assessment of human factors is very dependent on an assessor's understanding of risk. How does FDA educate its assessors regarding appropriate assessment of risk? How does this assessment differ based on the end user and context of use for the proposed product?
Yeah.
Thank you for the question. So in terms of assessment of the risk, we really look at it from a clinical standpoint. And when we look at the context of use, we're looking at who is using this product and what is the intended use for this product. So a product that is used emergently is going to have a different level of risk than one that's used chronically. And in terms of how we handle that as a division, we really try to communicate and to the best of our ability come to an understanding of what is considered a critical task and what is a potential other difference if we're identifying other difference. Our division looks at the comparative analyses, and we really try to discuss with each other. So for anything that may have where there may be some confusion about what the risk is.
Thank you. Next question, please.
We have a question for Dr. Wang. Is there a recommended allowable margin D in the FDA draft guidance?
There is not a recommended allowable margin in the guidance. So the 0.1 allowable margin was used in the example. But for each product, I think the allowable margin is a case-by-case situation. It's related to the indications and the risk for each critical task. So this allowable margin should be discussed with through the consultation with FDA, and the applicants recommended to provide the justifications and the evidence for their proposed allowable margin. Yeah, thank you.
Thank you. Online question, please.
Another question for Dr. Wang. What kinds of internal research is FDA conducting to mitigate regulatory burdens for CUHF studies?
Thank you for the question. So as I mentioned a little bit during the presentation, our group have been working on evaluating the performance of different potential alternative data analysis methods for the human factor studies. We are evaluating how well each method can control the type 1 error and type 2 errors through simulations. Yeah, thank you.
Can I add something?
Yes, please.
So, I mean, I think, Dr. Wang's group is doing a lot of work looking at some of the just different statistical approaches. And, we heard her talk that there's lots of different ways people can approach the studies. A point that I think I wanted to emphasize from Dr. Streets' talk was, we see lots of comparative analyses. We get lots of device products submitted. I think over 1,000 in the 6 years since we implemented the draft guidance. Less than 10% of those get to the point where the design difference is something that's unacceptable. So I think that's first something to note that, really we're talking about a small set of products compared to what we're getting in OGD. And the other thing I think it's worth mentioning is, we spend a lot of time today talking about the work that we're doing to support the development of the design of comparative use human factor studies, the statistical approaches and things. But I do want to emphasize, in the guidance, one thing that I think often gets missed is that, we're open to other approaches to justifying those differences. So if a comparative use human factor study sounds very burdensome, and that's something we've certainly heard from applicants, and as Dr. Ballard mentioned, that's part of why we're funding the research that we're funding. That doesn't necessarily mean that is the only way to validate and justify your other design difference. So I think that we're open to both considering alternative statistical methods, understanding the rationale for your justification of your non-inferiority margin, or if you think there's some other way you can justify why a design difference is acceptable under an ANDA and can be approvable. I think we're open to hearing those justifications and thinking about them. And I know Dr. Streets has mentioned, when we find those things in our assessment, sometimes we'll ask questions, ask for more information, more justification, and potentially changes, different things. And we do want to work with you to get these products to the market so the American public has access to them.
Thank you. Next question, please.
Next question, again for either Dr. Ballard or Dr. Streets. How does a generic drug sponsor submit a comparative threshold analysis for an approved product where a PFS is proposed, but the approved RLD is a vial, or that there are no approved generics with a PFS?
Okay, so this is something that we run into frequently. And I think when you're looking at that, you just have to consider what the design features are and what their purpose is. So while it may seem impossible to do a physical comparison of a prefilled syringe versus a vial, it's really important to just provide the information that compares them, and then the labeling as well, and then also, the task analysis. It's not impossible to do. You just have to submit the information and the comparison. So I can't think off the top of my head of a side-by-side, but if there's not an applicable side-by-side component to the comparison, then you can state that.
Yeah, I would just add that when we see this happen, and we've had it happen a couple of times where the RLD is a vial and they want to come in a prefilled syringe, or they want to come in an auto injector. And I think in that situation, you really do need to come in early and come in on the pre-ANDA side so that you can start to get our feedback early as you continue your device development, because there may be issues that we're aware of that we can help in guiding you through that process. But I think the main thing is going to be when you have that is the task analysis, because are you eliminating tasks? Are you making it easier for the end user? So you need to keep the end user in mind and the environment of use, because if the task analysis shows that there may be some advantages, we may be able to do that. There's certainly many products that were originally approved as a vial with a syringe, with or without a prefilled syringe as part of the kit, that have now come in as prefilled syringes or as auto injectors even. So it can be done, but we strongly recommend that you come to us early in the pre-ANDA space so that we can iron out those differences.
Okay, next question, please.
Another question for either of you, Dr. Streets or Dr. Ballard. Is it possible to address differences that are considered other through label changes and IFU and not only with device UI changes?
So you cannot label your way out of a difference. But again, we're not saying that every design difference needs to be changed. So again, there's additional information that you can provide to justify that the design difference is not likely to affect the clinical safety or, the clinical effect or the safety profile of the product when it's substituted for the RLD. So the change, you do not have to make the change, but you can't just change your labeling in order to make that okay.
And there's certainly data that can be obtained and extracted from other areas. Certainly what's out in the public arena in terms of real world data for known use errors and things like that, that can be brought in to show that, maybe justify your design difference. You can conduct your own study that may show that there's no difference, even if it's not a formal CUHF. So it's not necessarily that you have to do a CUHF if there's an other design difference and you don't really want to modify your design, or let's say you can't modify your design because of patent issues with the RLD. There are other data sources that you can use and bring in to help justify your design differences. And again, this is where it becomes very important if you're looking at something like this to come in both in the pre-ANDA space so that we can advise you early on, so that when you do get to the point of your ANDA submission, some of this has already been resolved and you know which way you have to go.
Okay, next online question, please.
This question came in addressed specifically for Dr. Ballard. If the RLD is not available in the proposed device, how does a generic drug applicant submit justification through a comparative threshold analysis?
Okay. What we usually recommend in this situation is you have to compare to the RLD, and that can be done. Certainly the labeling, you can usually get a copy of the RLD labeling even if the product is discontinued, and you can use that to do your side-by-side labeling. You can get instructions for use and use that to do your side-by-side labeling. Where the issue becomes stickier is when you're looking at the actual device itself, because the RLD may no longer be available, but usually there will be a reference standard that can be used not only for the drug product, but also for the device issue. And you can go ahead, and we have seen submissions where they have used the physical comparison to a reference standard as additional supplemental information to support the use of their generic, because the RLD is no longer available. But we still recommend that you try and make a best effort to get the RLD product, and if not, at least use the RLD product for the labeling and the task analysis.
Yeah, I would emphasize that part, that you should be starting with the RLD as much as you can find, as much information as you can find. Photos of the RLD, labeling, those sorts of things. And that some of these supportive or supplemental information from a reference standard or another generic version referencing the RLD can be part of that sort of other information that helps us evaluate and understand, is your proposed product going to be usable in the marketplace? Are people going to be able to use your drug product in place of the RLD, which, recognizing in this situation, the RLD is discontinued?
Okay, thank you. Next online question, please.
I have a question for Dr. Chong. How do you think the ongoing research work that OGD is supporting will help the development of generic combination products?
Thank you for that question. I think we've already heard a little bit about how some of this can be helpful. I think Dr. Ballard's initial response to the question around the taxonomy is one that potentially can be helpful, just to make sure we're all speaking the same language, acknowledging the initial questioner's framing of it is complex, but it is a starting point, and that might help get us to a place where it becomes more more second nature to do these kinds of assessments, and we're using the right terminology so that everyone is understanding when we're questioning a specific design difference, we know what we're talking about. So I think that's one place. I do think that, as Dr. Ballard mentioned in her presentation, designing comparative use human factors study, I think will be very educational and informative for us to understand where are the pitfalls, where are the challenges, what are the things that we can advise on to make the assessment of these other differences easier going forward in the future. And I can't speak to the statistics. I'm not that bright, but I think alternative methodologies for analysis of data, I think, is something we can certainly look at.
Yeah, and also I'd like to add that as we mentioned, as Dr. Ballard mentioned in her speech, that there are grants, so we are working on those things also. And for the methodology, actually, I want to tell you that FDA doesn't recommend any specific method for the analysis. So anything that is appropriate for the design is acceptable. Acceptable in the sense that if it works for that particular design, then we can accept that. So actually, the word or phrase alternative method actually is a misnomer in that sense, because there's an alternative to what? We don't have a prescribed method, basically. So anything that is acceptable, that is appropriate, may be acceptable. So I just wanted to stress on that. And you can, the key is to come to FDA early, so that if you have anything, any method, any method that is not widely used, if you want to use that, or if you have any innovative ideas, you can come and we can discuss that.
Yeah, thanks for the correction on the terminology there.
Okay, thank you. Next question, please.
One more question for Dr. Wang. Could you give an example of how non-parametric methods may be used for CUHF studies?
Thank you for the question. So for the non-parametric, I would like to give example like using the bootstrap-based methods. So bootstrap resampling may be used to through the resampling, it can be used to estimate some attributes of the target population for the statistical inferences. Then based on this methods, maybe the confidence interval of the non-inferiority test can be calculated. Yeah, this is a brief example. Thank you. Yeah, Somesh, if you want to add more.
Yeah, but whatever method, again, there may be other non-parametric methods, and but if you think of something new, just come to FDA and we can discuss that. Because Jenny just mentioned one particular one.
Next question. No more questions. All right. And let's anybody in the house has any question they're excited about Axon? No? Okay, let's thank our panel.